Thank you. Thanks, Greg. Thank you very much for having me. I appreciate being here to be able to share this technology with you. So Greg just said it's quite a long title. This is essentially 3 technologies that we disclosed as provisional patents that were kind of merged into a single PCT application originally. So I'm going to try to cover as much quickly as possible. Let's see. There we go. All right, so the primary researchers on this technology are myself, Christopher Buxa, who was a master's student in mechanical engineering with me, and Stephen Neary, an electrical engineering student that graduated about a year ago with me. We have been continuously funded on this research since we first developed the technology, which was about 2015, 2016. It started with the American Foundation for Surgery of the Hand, a small grant, and then a Women in STEM Faculty Development Award. And then we were funded by the NIH through a research supplement to promote diversity in health-related sciences, which ended last March. And then at that point, I started a new Research Accelerator Mechanism Award that's currently funding us through March of 2021. And I'm actively applying for other awards to continue supporting this work. So as I mentioned, the innovation is actually 3 parts. There's 2 device and essentially 1 method component to it. So the first is actually the platform itself. So it's a multimodal manufacturing platform specifically for advanced biofabrication of bone soft tissue scaffolds. And I'll explain what that is in a second. The second is the scaffold itself. So bone ligament and bone cartilage hierarchical functionally graded scaffolds. So basically the actual components that would go into the body. And then the last is the method of fabrication of these scaffolds, particularly using the platform that we've developed. So a little bit of background here. So ligaments basically are connective tissue that connect bones to other bones at joint locations. They're designed to withstand high tensile loads or pulling loads. They're made of tightly packed, aligned collagen primarily. The interface of ligament to bone is actually a functionally graded, it's both biochemically graded and biomechanically graded. So you see the picture on the right-hand side, you have what's called the enthesis region. That's the transition from ligament to bone. And in that transition region, you have unmineralized and mineralized fibrocartilage, which includes a variety of different cell types that come together in this region, as well as what's called extracellular matrix material, which is what gives you those biochemical and biomechanical gradients from one tissue type to the other. The structural function of this region is to reduce stress concentrations at that bone ligament interface, because as I mentioned, ligament is meant to withstand high tensile loads, and bones are designed to withstand high compressive loads, so pushing forces. So there are 2 primary challenges in orthopedics that we were looking to address with the technologies. The first is ligament rupture and reconstruction. So damaged ligaments currently, the way surgeons repair them is with something called ligament reconstruction with tendon interposition arthroplasty. And basically what that means is instead of using another ligament to repair a ruptured ligament, what they'll do is they'll actually harvest or remove a portion of a tendon that exists near the site of injury. So if you were in the wrist, as the picture on the left-hand side shows, you would use something called the flexor carpi radialis tendon that exists in that area, and you might use that to repair, let's say, a torn ligament between the scaphoid and the lunate bone here in the wrist. In the knee, you might use a patellar tendon autograft there, and then if you have kind of a multi-ligamentous injury on the knee, for instance, you might use a combination of multiple tendons to repair that. What are the problems with this? You have something called donor site morbidity, meaning you create a problem where there didn't exist one before, where you remove a portion of the tendon that was previously intact. Secondarily, you have a tendon which is similar but not exactly equivalent to a ligament mechanically. And then because of this, you tend to have a high failure rate, anywhere from 10 to 20%. This increases depending on when you've ruptured your particular ligament and how old essentially you live. So if you are a younger person who has a ligament tear or injury and you have a tendon reconstruction and you live a long life and you're very active, you're likely to need a secondary or tertiary surgery in order to repair that. The second challenge is much bigger. And we're talking about total joint replacement. Essentially right now, what's done when you need a total knee joint or total hip joint, you'll basically have a portion of your femur or your hip removed, or a portion of your femur or your tibia in the case of a knee, and they'll be replaced with either a metallic or a ceramic implant. And what we have in mind is something that would actually work to replace these metal or ceramic implants with actual human tissue, your native tissue, preferably, that can be regrown in the form, in the exact shape of your anatomy. So how did we want to do this? So we wanted to use additive manufacturing in order to design these scaffolds. But at the time when we came up with the idea, there didn't exist any additive manufacturing technology that could do this, that could create a composite scaffold that could create a structure that in one end was could resist high compressive loads, on the other end could resist high tensile loads, that was fully integrated from one tissue type to the other, such that there was no failure that existed in between the two, so no high stress concentrations, the same way bone and ligament join together naturally. And so, but we did know that there existed a few types of technologies that could do something similar, but not exactly at the same time. So we know that 3D printing can print microscale fibers. You can print something using patient-specific geometry with MRI or CT data. And we know that the three-dimensional structures that are printed can resist high compressive loads the same way that bone can. On the other hand, we know that 3D printing is not always so great when trying to make something that can resist high tensile loads. But there did exist other technologies, one being something called electrospinning, that could actually deposit nanoscale fibers that could be tightly packed to promote cell attachment, cell adhesion, growth, things like that. And if aligned properly, could resist high tensile loads. And so what you see there on the bottom is kind of a picture of what looks like kind of a spaghetti bowl deposition of nanoscale fibers using a standard electrospinning technique. We wanted to be able to control this architecture very specifically. And so we knew that some combination of 3D printing and something like electrospinning, if we could bring this together into a single platform, could work well for us for the technology. And so we created this system. We actually called it our eSpin printer, that could do hybrid additive manufacturing. And I'm focusing specifically on these two technologies that were merged into one, kind of merged into one, but there are actually a lot of other things that our system does. But what you see here is the first prototype that we actually made in our lab. It's a working prototype that existed early on. This is just a single tool head that was a 3D printer, and a single tool head that was an electrospinner with a common platform underneath that could serve as an electrically conductive surface for the electrospinning side and could be a build platform for the 3D printing side. But it was integrated to be used such that we could make a composite structure where both phases of the material or multiple phases of the material were integrated fully. And so on the right-hand side, you see what kind of a generic design of this might look like. So the red ends of this structure are the two bony ends. The lines that you see in the middle, that's our ligamentous fibers. And then you see these kind of transitional regions, the mineralized fiber cartilage, the unmineralized fiber cartilage, where in these boxes, per se, this is very generic, so they wouldn't exactly look like this, but in these boxes, you could include various cell types. So you could have, let's say, cartilage type cells that would exist into the fiber cartilage region. You could have osteoblasts in the bony phase. You could have fibroblasts in the ligament phase, and so forth. And then you can actually include various other growth factors and other things that you needed in order to create a functional scaffold. Early on, we completed our feasibility testing against benchmarks. So we wanted to prove that just like any standard bioprinter, it could actually print gels, specifically hydrogels that could be used to print anything that you wanted. We also wanted to verify that we could integrate different bio inks or different types of materials to create common or single structures. That's what you see in these little videos here that are playing. We showed that we could do architectural gradients. You saw the small end, they go a little bit bigger, they go a little bit bigger as they move down here. And the first video actually showed material gradients from one material type to the other. On the bottom there, you see scanning electron microscope images of the electrospun. It's what's called almost direct right electrospinning, where we can control the electrospun fibers to create architectures that are ideal for us. So either uniaxial fibers, we can have this 90-degree crosshatch, we can have these 45-degree angles. We can basically anything you can do with a 3D printer, we can do so with this direct right electrospinning system here. And that was important to us because we needed to build our first scaffold and prove that we could actually make this fully integrated from a ligament phase to a bone phase and everything in between. And so I'm running out of time here. So the picture in the middle just shows you, this is essentially half of that picture I showed you a couple of slides ago. That was the pink and red colors. So what you see is the middle here. This is the ligament, the pure ligamentous phase with the varied architecture that we wanted with the fibers. On this end, you see the pure bony phase where we could include various things, the various cell types and growth factors into these pores. And then you see this transitional region where we have both ligamentous and bone in this kind of mineralized and unmineralized fiber cartilage region. And we tested these mechanically to make sure that we were seeing that they were fully integrated fibers into this bone phase. And so this picture on the top right is what it looked like in that ligament phase before we did anything to the scaffold. Then we mechanically loaded that scaffold in tension. And that's what you see on the bottom right there. Those fibers did not pull out. They were fully integrated into the bony transition region. And you saw failures that existed exclusively in that ligament phase, which was essentially kind of our weakest link in the fibers or in the composite. So for need market potential, what we know is that over the next 5 to 7 years, the bioprinting market is estimated to grow at about 15%. It's anticipated to grow over 4.7 billion by 2025. This is some research that's been done by this group here. I put a few pictures of some existing technologies that would essentially either be competitors, potentially partners, we can work with. The one we've been keeping the closest eye on is this RegenHU here on the left-hand side. They're based out of Switzerland. They are currently the only system that we're aware of that can do kind of this hybrid biomanufacturing specifically for tissue scaffolds. Everything else that you see here, this one on the right-hand side, has other features like laser-assisted microvalve and extrusion bioprinting. You can see that they're selling for over $200,000 a piece. Moving into kind of the lower price range, we've got an EnvisionTech system. This is out of Germany. It sells for about $150,000. And then something a little bit more reasonable, the Cellink Bio is purely a bioprinter, and it sells for about $30,000 in the United States. We made our hybrid system, just so you know, for about $10,000. And that is like well below the cost of just a standard bioprinter. And if you add the electrospinning component to itself, electrospinners are selling for about $50,000 to $100,000 as standalone systems. So our current status is that we do have 2 working lab prototypes, and we have another one in development that we're trying to improve the hardware components that we're using so that we have nice interchangeable tool heads. We have done some research on the best materials to use for our bone, ligament, and our interface phases, and we have completed laboratory-based testing of all of these materials, both biochemically and biomechanically. We're currently working on new materials development that we believe will improve the bone and the ligament phases mechanically and chemically. And we'd like to do some additional architectural analysis of the scaffolds so that we can continue to improve mechanical integrity of them. The funding that I currently have right now was for preliminary testing of in vivo animal studies for our scaffolds specifically. And really what we're looking for is to find an industry partner that we might be able to work with to obtain some SBIR or STTR funding. And that's everything.
Thank you, Christina. That's great. If there are any questions, send them in the chat. I have a question. Have you done a comparison between how this holds up compared to natural tissue? Or do you?
Yeah, actually, that's our benchmark. That's what we're comparing all of our tissue to. So my lab is a biomechanics lab, and so we work primarily with human tissue. And so all of our bone phase is actually being compared against actual cancellous bone that we harvest from our cadaver material, or for our cadaver specimens. And the same thing for ligament. We typically compare it to the scapholunate ligament in the wrist. And that's actually why our first source of funding came from the the Hand Society, because they were really interested, or they are really interested in our technology specifically for problems with ligament injuries in the wrist.
One other question. If you were going to move ahead with, there's many applications for this. Is there one particular, indication or, injury type that you would try to focus on as your first attempt at proving?
Yeah. I think starting small is probably smart. And so we have been focusing specifically on that scapholunate ligament injury, and not only because there's a high prevalence of that injury in people, let's say, who fall on an outstretched hand, or, who have tears because of work-related injuries, but it's a complex ligament. It's not just a straight ligament. It's actually a U-shaped ligament. So it allows us to show that we can make a more complex architectural design with ours. But I think that it could have a big impact, and it's a small joint. So we want to start to make sure that we don't have any problems with, or that we can maintain biocompatibility, and we don't have a lot of rejection or anything that you might see in a large joint.
We got one other question. What kind of type or industry partner would you be looking for?
It would be great if there was somebody who already had a 3D printing experience, a 3D printing company that knew about the hardware and software of 3D printers, because ours is not very different. We use a commercially available software to control our system. We would like to create custom software for ours. So if somebody knew how to work with or how to develop software for 3D printers, that'd be great. And then, like I said, hardware, we're working with a lot of really generic hardware that we have available to us in the lab, but there are, I think we can improve the design quite a bit.
Okay, thanks a lot, Christina.